I'm a chemical engineer. I do biology. Well, okay, I'm a professor. I don't do much anymore but talk and write and stuff, right? But my lab is very much focused on biology. But we do our work from the lens of folks who are trained to think about chemistry. And I like to use this figure as a way of putting things into context. And I like Levi's introduction. It was conserving resources, protecting nature. Did I get it right? This is one of the resources that we've been using way too much without thinking about both the downstream impacts and the fact that it's finite. So one of the first classes that we teach in chemical engineering is about material and energy balances. And so if we think about this from that perspective, most of, this is by the way what happens to a barrel of crude oil in the US, most of it is something that we're familiar with, which is using the material there for energy. That's the fuels part of the pie, and we're all familiar with the work to electrify transportation to be able to reduce greenhouse gas emissions associated with that. What my group has been more interested in is thinking about the part that is a much smaller segment when it comes to volume or mass, but has a very significant value, and that's using the part of crude oil where we use the mass for mass. So it's really about making stuff. When I tell first year students what is chemical engineering, like it's really making stuff, right? What are all the principles and processes around how you make stuff? These are things that we need for life and for quality of life. And so to be able to do the things that we do. So it's that, and there's just several examples there of different materials that actually come from petroleum-derived resources. So what my group has been focused on since I arrived at MIT really is, I'm borrowing this phrase from a colleague of mine, Brad Olson, who said, the problem is we need a paradigm shift in engineering, and in particularly for how we produce the materials and chemicals that we use, where instead of thinking about hydrocarbons as the input, we need to think about what are the renewable, sustainable resources that we can use. And so to have a much more carbohydrate-focused economy rather than a hydrocarbon-focused economy. And the biofuels, which Selena mentioned in her talk as well, was one of the first places where we really started to hear how we could use sustainably derived resources, feedstocks, to be able to displace petroleum. But we've really been much more interested in the biochemicals and materials space. And this is something I don't have to say to this group, which is that we know biology is very capable of doing chemistry. I always like to use this figure because I graduated 30 years ago, and as a chemical engineer, that's what we were told a cell looked like. Don't worry about any other stuff. It's just a thing, right? And much of my training as an undergraduate was about the processes around biology. But of course, we know that a cell is actually just a collection of a lot of chemical reactions. And so the principles in terms of how we think about how do we design a process to make chemicals in a chemical plant, we think about this in the context of that cell being a tiny chemical factory. So we know that we have to provide a number of inputs. Usually that's a carbohydrate-based sugar or carbohydrate-based feedstock. Oftentimes you need oxygen and other nutrients. And then there's a bunch of stuff that we can make, from pharmaceuticals to amino acids to specialty chemicals. What my group has been focusing on is how do we actually think about the fact that biology can do chemistry very well, but not necessarily limit ourselves to what we know that biology has made. So all of the examples I have there of molecules are natural products, but the question is, can we apply the same principles that chemists use to be able to synthesize new molecules in a flask towards designing biological systems that can synthesize these new molecules? So I'm not going to give a super technical talk because I only have 10 to 15 minutes, and you guys wouldn't want to hear it anyway. It's okay. You can admit it. Okay? But I want to walk you through a few examples. There's three molecules I'm going to show you here, and I'm going to come back to glucaric acid in a second, where we were able to design a pathway to make this using 3 genes from 3 different organisms. Doesn't sound like that big of a deal. The organisms were a yeast, a mouse, and a bacteria. Right? So the real power that we have is that biology is fungible in the sense that if we know what the DNA is that encodes for the enzymes that we want, we can synthesize it now. It used to be we'd have to obtain sources of cDNA for a mouse, for example, or we would get microbial samples and have to PCR. Now all we need is the sequence, the digital information, and we can get access to that material. So that allows us to think about how we can be more complex. So how do we make a compound where now we're taking multiple genes from still 3 organisms, or where we're taking even more genes from even more organisms. And the 3 molecules that are there, I'm going to come back to glucaric acid in a second. The one in the middle is a precursor to pharmaceutical, and the one at the bottom was actually our attempt to design the perfect biofuel molecule. Spoiler alert, there is no such thing as the perfect biofuel molecule. But this was sort of our attempt based on what we heard from a sponsor in terms of, if you could make any molecule you want it and have it serve as a biofuel, what would it look like? And so that's sort of the closest that we got. What we are focused on as we do this work, yes, there's the intellectual challenge that's there, but we want to be able to do something that's economical, right? So being able to produce a compound where we'll be able to compete in the marketplace. But those last 2 are what are really important to me personally, and certainly what drives the work that the students and postdocs in my lab do, which is that we are really interested in sustainability. We want to be able to contribute to solutions rather than continuing with problems related to sustainability. And so we're always thinking about what the environmental footprint is. So I just want to walk you in the last few minutes through 3 examples of different ways that we've done this. And I'm actually going to talk about collaborative work as well, just so that you have a sense of the kind of things that we're doing at MIT to address these climate and sustainability problems. The first one here, glucaric acid, that's a molecule, one of the first ones that we started working on. We actually licensed this technology to a company that I co-founded called Calion. And to be frank, when we started, what we could find in the literature in terms of where it might be useful, it turns out it's in Adderall. We can talk about that later. But there was a lot of research that had been done on trying to replace phosphates in the environment. Okay, so where does that have a role here? So this is a cooling tower. Again, I'm a chemical engineer. This is a piece of equipment that exists in really all major manufacturing plants. And essentially, it's a very large facility where, as you bring in warmer water, you actually drop it from a high distance, and it causes evaporation. It cools it down, and now you can pass that cooler water into other parts of the manufacturing facility. Well, you actually have to treat that water, especially because it's now processed water. And the traditional way to do this is by using phosphates. The problem with it is that a large amount of the water is recycled, but some amount of it is discharged into the environment. And if you have large increases in the amount of phosphate in the environment, it's typically limiting for algae. If you suddenly provide a lot of phosphate, the algae blooms, it blocks the light, everything under it dies, right? And you create these very severe environmental problems. So working with customers, what we've seen is that you can displace now this molecule, or replace rather this molecule with glucaric acid, and it has the benefit of both preventing, this is called eutrophication, it prevents these algal blooms, but it turns out that it also prevents corrosion. Why is that important? Well, corrosion in these systems actually leads to lower efficiencies. Lower efficiencies actually means you use more energy. More energy, as Selena just taught us, means more greenhouse gas emissions. So back of the envelope calculations, we have determined that our best estimate is that if you could get rid of all the phosphates, you actually reduce greenhouse gas emissions, or CO2, greenhouse gas emissions by the equivalent of 230 million metric tons of CO2 per year. And the increased efficiencies that you get actually result in cost savings of about $60 billion. The point I want to make here is that we had no idea that all these other benefits could happen as we started this project, but it does emphasize the fact, again, as Selena was saying, there are all these interconnected features or characteristics that we have to think about in terms of not just where materials are coming from, but what is their impact downstream once they use where they're actually being, once they leave where they're being used. The second example I want to go over is agriculture, that was also listed in Selena's slide as well. This is actually part of what is known as the MIT Climate Grand Challenges. These were projects that were launched about 2.5 years ago, and the lead PI here is a colleague of mine, Chris Voigt, who is department head for biological engineering. And so you can see this is a pie chart from the US, from globally where greenhouse emissions come from. About 24% are related to agriculture. And in the US, about 29% of energy use is for fertilizer. Okay, so agriculture necessary, right? It's not optional to be able to feed ourselves. And so the question is, how do we actually address more sustainable agriculture? So one of the things that's interesting is that if we look to nature, nature usually has suggestions for things that we could do to make life a little bit better. And in particular, legumes are a category of plants that actually get their nitrogen from the air. They're able to fix nitrogen, not the plants being able to do this directly, but rather there are symbiotic bacteria that live in the soil. Those bacteria can fix the nitrogen and convert it into a usable form, so into ammonia. And then the plants are making the carbohydrates that will feed the bacteria. The bacteria are making the ammonia, which will feed the plants. That's true for legumes. It's not true for cereal crops. And it turns out we have a much greater need for cereal crops in terms of meeting nutritional demand than we do for the legumes. So the idea here is to be able to engineer now bacteria that will associate with cereal crops for this ability to fix nitrogen from the air and therefore be able to reduce greenhouse gas emissions. And so a startup that came out of Chris's lab and that was founded actually when he was still in California, but has leveraged IP from MIT called Pivot Bio is doing this, where they are selling microbes to farmers that they can plant with or put down with the seeds. It actually dramatically reduces their dependence on externally supplied nitrogen. Well, so that's great, but we like to think big at MIT, right? So the project that the Climate Grand Challenges is focused on has two different parts of it. One is to actually be able to engineer these crops for direct fixation, right? So to say, well, take that nitrogen fixing ability and move it from the microbes into the plants. But then you can have nitrogen provide either in the form of ammonia or in the form of urea. Urea contains carbon, and so it turns out you can enhance carbon fixation and reduce the demand for externally supplied fertilizer at the same time. Very hard problem, right? This is not a problem that's going to be solved by the next time one of these symposia exists. But it's a way of thinking about if we really needed to address this on a larger scale, let's set some targets, let's set some goals and figure out what biology can really do. And the last example I want to give you is a collaboration I've had with Brad Olson, who I mentioned earlier, who's a colleague in chemical engineering, and Desiree Plata in civil and environmental engineering. And it's about plastics, right? So the Monarch system minimizes plastic use. We still need plastics, and so the question here is, can we actually attack this problem of all these plastics being produced, most of which end up in landfills or the environment, and it doesn't actually go anywhere? So what we've been working on through a series of collaborative projects is thinking about this whole process, starting with where the monomers come from. So can we have now, rather than fossil-derived monomers, can we have monomers that are derived from biology in the same way that I talk about using biology to synthesize chemicals, to then be able to produce materials where we know how they perform, so we can replace like with like in terms of what the function is. But the important part here is to figure out what happens now if you can't recycle it, if it's dropped in the sidewalk, if it washes off into a waterway, or if it ends up in the soil. So what we've been doing is working on developing these high-throughput biodegradation assays to be able to really understand what happens in the ecosystem in a way that we can reduce the screening time from months to a few weeks, and then also be able to adapt it to better mimic what happens in the environment. So the longer-term goal here is to make a material that you need from a sustainable source, but know that wherever it goes in the environment, if you can't recycle it, as shown on that far right side, that you can at least know that it's going to be degradable in the environment and it won't continue to contribute harmful waste. So hopefully you've gotten a good sense of how we think about using the power of biology in order to do chemistry. I love this was created about 15 years ago now, and I just love it because it's how we now see cells, right? Not as this thing where there's nothing inside of it, but really as a way to be able to transform how we access the materials that we need. So with that, I thank you for your attention, and I'm happy to pass it on.